Hello everyone, a very good evening. I welcome you all to the National Workshop on CSIR-UGC-NET-JRF LS Preparation in Life Sciences organized by Botany for You. Today is the first session of your day 5 and in this session we will be studying about the cell division and the cell cycle. So first let me introduce about myself. I am Assistant Professor KIRUBHA Pauldas. I passed my B.Sc and M.Sc degree from the University of Mumbai and then qualified CSIR-NET and M.Sc, and currently I am working as an assistant professor in the Department of Microbiology of SICES Degree College. So now let's begin with the lesson. In this particular session, we will be studying about the overview of the cell cycle, the 4 different phases of the cell cycle, stages of mitosis, checkpoint control of the cell cycle, the regulators of the cell cycle. We will include discussion on both the positive regulators, that is your Cyclin CDK, as well as the negative regulators PRB, P53 and P21. Now what is a cell cycle? Cell cycle is a life cycle of the cell, wherein one cell can divide into 2 identical daughter cells. It is an orderly sequence of events which takes place inside a cell, and in this particular events, the cell's genome as well as its cytoplasm duplicates and then distributed to the 2 daughter cells. For this fair distribution, 2 of the processes are very important. One is your DNA must be faithfully replicated, and secondly, the replicated chromosomes, it must be segregated into the 2 daughter cells. Then only your daughter cells will be genetically identical. Now during this process, not only the genome is duplicated, but the cell also double its mass. It means it grow itself as well as it's duplicate all their cytoplasmic organelles. Thus we can see cell cycle, it is a complex set of cytoplasmic as well as nuclear process which has to be coordinated with one another. Now let us understand what are the different phases of the cell cycle. The cell cycle is divided into majorly 2 phases. One is your interphase, which comprises of the G1 phase, the S phase and the G2 phase, and then the M phase. Let us understand in detail what all major processes takes place in this particular cell cycle. First, in the G1 phase, G1 means gap, it also means growth. So this particular phase is a gap phase between your M and S phase. As I mentioned, there are 2 major processes that take place in each cell cycle. One is your DNA replication and the second one is the cell division, where your genome as well as the cellular components are distributed equally to the 2 daughter cells. So of that, the DNA replication is going to take place after the G1 phase. So before the cell moves on to that particular phase, the cell is going to prepare all the components required for the process, and that will take place in the G1 phase. During the G1 phase, if the cell do not receive proper external signals as well as internal signals for cell division, the cell can go to the G0 phase, which is the resting phase. In this particular phase, cell cycle is halted permanently. And the one example is your neurons, which is permanently in the G0 phase and it is not going to divide further. After G1 phase comes your S phase, which is called as the synthesis phase, and here your actual DNA replication takes place. So the amount of DNA in a cell which is in the S phase will be double as compared to the cell in the G1 phase. After your S phase, you have the second gap phase which is between the S and the M phase, that is your G2 phase. The most important one, that is your cell division or segregation, which is also called as mitosis, which is going to take place in the M phase. So beforehand, in this particular growth phase or the gap phase, the cell is going to prepare all the proteins, enzymes, cofactors required for the M phase. And by the end of G2 phase, the cell is ready with all the components required for the M phase. So what is going to happen in the M phase? In the M phase, the actual nuclear division takes place, which is called as karyokinesis. And by the end of the M phase, we have a cytoplasmic division where your cytoplasmic organelles are getting divided, called as cytokinesis. Before moving ahead, I would like to tell you all that the cell cycle is unidirectional. It means it is always going to go from G1, S, G2 and then to M. From M, it is never going to come back to G2. We call this as a cycle. Cycle means what? Whose start point will be its end point. So, a cell who starts the cell cycle in the G1 phase is going to go to S, G2, M and then again going to return back to the G1 phase. Now, let us understand the stages of this interphase in a little bit more detail. As we can see over here, this is a cell which is just came out of your M phase. So, it is now going to be at your G1 phase. In the G1 phase, the cell is going to double its mass. It is going to grow as well as it is going to produce the molecules, proteins, enzymes required for DNA replication. As we can see over here, I have shown you the nucleus of the cell as well as its genome. The genome is now in the form of a chromatin because that is the form which is required for DNA replication. Along with that, you can see over here the centrosomes. Now the centrosomes play a very important role in the M phase. The cell is also going to possess all the other cellular organelles. If you say if this is an eukaryotic cell, then it is definitely going to contain organelles like mitochondria, chloroplast, endoplasmic reticulum, Golgi, etc. But since we are not right now interested in them, you can see they are not visible in the cell. Fair enough? Now let's see a cell which is in the G1 phase. It is now moving to the S phase. S phase, we said the DNA is going to replicate it. As we can see here, a DNA, a new copy has already formed. Take a note here that along with the DNA, your centrosomes are also duplicated. Right? So after the S phase, the cell moves to the G2 phase, and in the G2 phase, you can see the cell has again grown in size. So it is a growth phase, and during this phase, the cell is also going to produce all the components required for the M phase. Now let us turn our attention to the M phase. As discussed, M phase includes two major events. One is a nuclear division called as karyokinesis, and second one is the cytoplasmic division which is called as the cytokinesis. Now this nuclear division is divided into four stages. One is your prophase, then metaphase, then anaphase and then telophase. Now they are going to occur in a sequential manner. Now why they are named so? In Greek, pro means before. Since prophase is going to start first, hence it is called by that particular name. Meta means middle, ana means after and telo means the end. That's how the stages of M phase got its name. Let us understand what are the major events that takes place in these particular phases. This is a cell which was in the G2 phase and it is now got a signal to go to the M phase. Now during the M phase, the first phase is called as the prophase. Now what happens in the prophase? Here, the chromosome which is made of two genetically identical chromatids which are joined by a centromere. Usually during the DNA replication, the genetic material is loosely packed as chromatin. But for mitosis, we need the DNA to be very tightly packed. And that's what happens in your prophase. Your DNA becomes condensed and then the both copies of the DNA are held together by the centromere, and we call these copies of the same chromosome to be sister chromatids. Make note here that there are two important proteins which are involved in condensing your DNA. One is a protein called as condensin, which actually condenses your DNA, and the second one is known as cohesin. Now, cohesin proteins are those which helps for your sister chromatids to remain together until the end of the metaphase. Another major event that occurs in prophase is, you can see here, the nuclear membrane has started to disappear because now the chromosomes have to come out of the nucleus to the cytoplasm. They can undergo the further process. Also, in the prophase, your two centrosomes start to move towards opposite poles. Now let's go to the metaphase. In the metaphase, we can see your chromosomes, which are now in the cytoplasm, have aligned themselves at the equatorial plane of the cell. And then you can see something amazing also over here. We can find the centrosomes on the two opposite poles, and there are something thread-like structures which are called as spindle fibers, which are arising from this particular centrosomes, and they are attaching to your chromosomes at the centromere. These particular spindle fibers are structures made from microtubules, which helps in the organization and the arrangement of your chromosomes. Now there are three types of spindle fibers. One which is called as the kinetochore spindle fiber, which attaches to the centromere of your chromosome, and the second one is called as your polar spindle fiber over here, which attaches to itself. This particular spindle fiber originates from this centrosome, and this one comes from this particular centrosome, and both of them are getting associated with each other. These are polar microtubules. Third, we have, it is called as astral microtubules or astral spindle fibers, which originate from these particular ones, the small ones we can see, which originate from your centrosomes. Now all three types of spindle fibers or microtubules play a very important role to separate your sister chromatids equally and then taking them towards the poles. These particular spindle fibers are named as plus and minus. The minus end are the one which are attached to your centrosomes, and the plus ends starts to add your tubulin proteins towards itself, and it starts growing or elongating itself. Now once this particular arrangement is finished, the cell will progress to the anaphase. Now what is going to happen in the anaphase? The sister chromatids are going to get pulled to opposite end of the cell. Here the spindle fibers will contract. So it is going to pull your chromosome towards the pole along with themselves. As well, here we can see these spindle fibers which have not attached to your chromosomes, they will elongate the cell to prepare the cell for cell division. Now after that, your cell prepares for the telophase. In this phase, the cell has elongated and is nearly finished dividing. We can see the features which was lost after the prophase start appearing. Like we can see here, our nuclear membrane started appearing, and then our chromosomes have again turned back to a chromatin state, and we can see there is also a constriction at the ends of the cell where your cell is going to get constricted and divide. And also we can see that the mitotic spindle fibers have now broken down. This is the end of your karyokinesis. Now the last step is your cytokinesis. So this is the division of the cytoplasm to form the two new cells. Now to separate the two cells, a ring of protein which is called as the actin ring, this will pinch the cytoplasm along a crease which is known as your cleavage furrow. And then this will split your cytoplasm equally between the two cells, and your two cells are released at individual cells. Now these two cells which are released after cytokinesis, these are again will enter the G1 phase. We have understood the cell cycle and the major events. Now let us move towards the regulation of the cell cycle. The regulation of cell cycle is with the help of the checkpoints. Now what are checkpoints? We have seen that police usually put a barricade on the road and they check all the vehicles. Something is going to happen also in your cell cycle. The cell cycle which starts at the G1 phase is going to encounter the first checkpoint called as the G1S checkpoint, which is found at the transition between your G1 phase and your S phase. Now what all events are going to be checked in this checkpoint? The cell is going to check for the cell size, the nutrient availability, as well as molecular signals. Now molecular signals are very important because these are going to tell the cell that now it is ready to move to the next phase. Another important factor is the DNA integrity. It is going to check if the DNA is not damaged or if it is okay to move ahead, because cell cannot take the risk of replicating a damaged DNA. So if all these conditions are fulfilled, then this particular checkpoint is lifted up and the cell can move towards the S phase. It can replicate its DNA, then it can go to the G2 phase. But at the end of the G2 phase, the cell again encounters a second checkpoint, which is called as the G2M checkpoint. And in this checkpoint, the cell is going to check for DNA replication, whether it is properly completed, as well as the DNA integrity. We know that during DNA replication, there is a possibility of mutations in the cell. So if there is any mutation, like if there is any base addition, deletion, frame shift, any mutation, the cell cannot pass on this damaged DNA to the daughter cells. So cell is going to check in this checkpoint, and if it finds that there is issue, the cell cycle will stop at G2 phase. It will give the signal to go for the repair pathway. If repair is not possible, the cell can move towards the apoptosis, wherein the cell will destroy itself. But if all these conditions are met, then the cell can move ahead to the M phase. At the M phase, there is one more checkpoint called as the spindle checkpoint. This checkpoint is between the transition from the metaphase to anaphase. It is going to check whether your chromosomes are aligned properly at the equatorial plane, as well as are they all properly attached to the spindle fibers. If not, there is a possibility of unequal distribution of your DNA. And if it encounters such problems, that mitosis is going to stop here, and then it will only continue when that particular issue is resolved. The cell is then going to move ahead and then finish its M phase and creates its daughter cell. So that is how this particular checkpoint works. It is going to ensure that your every cell cycle takes place in a coordinated manner and also in a proper way. Now let us learn, so how are these checkpoints regulated? There are some proteins called as cell cycle regulators. We have two types of regulators. One is your positive regulator and another one is your negative regulator. The positive regulated, these are proteins, they get activated and they initiate the cell cycle. So if this proteins, let's say protein X, it is a positive regulator. So if it gets activated, then it is going to allow the cell cycle to progress from one phase to the another phase. Whereas your negative regulator, if these particular proteins gets activated, they will halt the cell cycle. Remember one thing over here, that even though negative regulators are halting the cell cycle, they are going to work for the benefit of the cell. So they will halt the cell cycle only when the cell encounters some damage or cell encounters some problems at the checkpoint. The example of positive regulator is your Cyclin CTK, and the example of a negative regulator is your PRB, P53 and P21 proteins. Now let's have a very quick review about how exactly this positive and the negative regulators are going to work. So positive regulator is your Cyclin and CTK. So these are the two molecules. One is your Cyclin. This protein is going to go through a series of synthesis and degradation, hence the name Cyclin. Well, it is going to attach to a protein called as CTK. CTK means cyclin dependent kinase. So it is actually a kinase enzyme. We all know that kinase enzyme transfer a phosphate group from a high energy molecule to a target molecule. So that is a function of your Cyclin CTK. Remember one thing here, that CTK is always present in the cell at all circumstances, all throughout the different phases of the cell cycle. But cyclins are not. There are different type of cyclin which act at each phase of the cell cycle, and they are the G1 cyclin, which is going to act at your G1 phase, G1S cyclin, which is active at your transition between your G1 and S phase. Then we have S cyclin, which is active at your S phase, and M cyclin, which is going to work in your M phase. So these particular cyclin molecules, they will be synthesized only during that particular phases, and when they are synthesized, the CTK can easily bind to them, and once your cyclin CTK protein is activated, it can allow the cell to move from one phase to the another. Now in vertebrates, these cyclins have some particular names, and these are, the G1 cyclin is known as cyclin D, it attaches to the CDK 4 and 6, while G1S cyclin is called as your cyclin E attaching to the CDK 2. S cyclin is also called as cyclin A attached to the CDK 1 or CDK 2, and the M cyclin is called as cyclin B, which attaches to the CDK 1. So these are the names of the cyclin CDK complexes which are found in vertebrates. Sometimes the cyclins are also called as S-CDK, M-CDK. It means S-cyclin along with its CDK. Okay. So once a particular cell have to move from one phase to another, since it is a positive regulator, cell will synthesize them and cyclin will bind to CDK. They will get activated and they will allow the cell to progress. I hope you have understood the function of positive regulator. Now let us understand about how exactly your negative regulator is going to work. These negative regulators are actually gene products of the tumor suppressor genes, and when they are mutated, they can cause tumor. Now let's see the function of this PRB. There is a protein called as E2F. Now this E2F is a transcription factor, meaning it can bind to the promoter region and allow your RNA polymerase to come and bind properly at the promoter sequence so that it can transcribe your downstream gene. So this E2F is going to bind to the promoter here and allow the transcription of cyclin A. Now what is a cyclin A? Your S cyclin, the cyclin which is active at your S phase. Now but it cannot do so because your PRB, which is now active, is actually binding your E2F together. So when it is bound with each other, your E2F cannot perform its function. But once it becomes inactive, and then your PRB cannot hold your E2F anymore, and hence it is released, and now it can allow the transcription of your cyclin A and allow the cell to move towards the S phase. So we can see the PRB, when it was active, it was blocking the cell cycle, right? Once it got inactivated by phosphorylation, only then it could initiate the cell cycle. So it is important, as we saw in G1S checkpoint, that the cell has to stop there and the checkpoint verifies various factors and only it gives a green signal, and then your PRB becomes inactive and then your cyclin is synthesized and then the cell can move towards the S phase. So that's how exactly your negative regulators work. Now let's understand about this P53. P53 is again the transcription factor, but it is always bind to its negative regulator called as MDM2, and once MDM2 finds P53, it is going to destroy P53. But in some cases, this particular P53 becomes activated by phosphorylation, and since it is a transcription factor, it is going to bind to your promoter, allow your RNA polymerase to bind to the promoter sequence and transcribe the downstream gene, which is the gene for the P21 protein. Now, this P21 protein is actually a CDK inhibitor protein. It means it is going to bind to the active form of cyclin CDK complexes and block the cell cycle. Because if active form of cyclin CDK molecules are present, the cell cycle can proceed further to the next phase. But if the cell encounters any problems or if it encounters any DNA damage, what it will do, it will activate your P53 by phosphorylation and allow the transcription of P21, which is a CDK inhibitor. It is going to inhibit your SCDK, it will inhibit your G1S CDK so that the cell cannot proceed to the S phase. So that is how your negative regulators work. When they get activated, they are going to prevent the progression of cell cycle or they are going to halt the cell cycle. I hope so you have understood all these particular concepts that we have discussed in this particular session. We started with a simple understanding of the cell cycle and then we move on to the different phases which occurs during the interphase as well as during your M phase, and then we understood the regulation of cell cycle as well as the roles of positive and the negative regulators in the cell cycle. So thank you all for your patient listening. So if you have any questions or queries, you can comment below this video or else you can ask in the Telegram group. And if you want to learn in detail about all these particular processes, you can visit our YouTube channel Learn Microbiology. Search in the YouTube and you can find the channel with this particular logo. Do subscribe to the channel so that you will be notified whenever I post a new video. As well as you can connect with me on Telegram. You can find this particular Telegram channel called as Learn Microbiology and subscribe to that channel. Thank you all.